Hi, everyone. I'm really happy to be here. Today, I'm going to talk about the project we started relatively recently. I'm going to talk about the brief idea, the analysis framework, and show some very preliminary results. It's about estimating social genetic effects on Alzheimer's disease. So I don't have to convince people in the room that GWAS, now we think it's more complicated than a lot of people initially think. So one particularly interesting thing people identified relatively recently is that parental genotype can directly cause changes in children's phenotype, as illustrated in some of the landmark papers in recent years. So then when you run a standard genome-wide association study, the effect size you capture is going to be a mixture of these direct effects coming from your genotype to your phenotype, and also this genetic nurture effect coming from your parents' genotype directly to your phenotype. So then we started thinking about, can you actually perform GWAS to estimate effect size based on this direct and indirect pathway? Turns out you can do that. If you actually have a lot of trios, this is a trivial problem. So basically all you need to do is to regress someone's phenotype, for example, educational attainment, on both parents' genotype and your genotype. You need to do a multiple regression instead of just running a marginal regression. And if you have a lot of trios, this type of multiple regression actually will give you very good estimates about the direct and indirect effect sizes. The problem is just nobody has that many trios because we're still talking about very tiny effect sizes, so you need a large number of trios to do this. So the approach we thought of is that you basically need to run 3 sets of GWAS. The first GWAS is a standard one. You regress everybody's phenotype on their own genotype. And then the second GWAS, you regress children's phenotype on mother's genotype. The third one is similar to that. You regress children's phenotype on father's genotype. If you are able to run 3 GWAS like that, basically you can use linear combination to reconstruct the actual parameters you're interested in. The parameters are direct effect sizes, maternal and paternal indirect effect sizes. So it's a simple linear algebra. The advantage of this approach really is just that instead of having trios, you basically need to go to, say, UK Biobank and ask about their children's phenotype. Because, for example, in the second GWAS here, you do not need children's genotype. You just need to ask all the mother's children's education. So that's the advantage. You only need to collect the phenotype. So then after we got this project wrapped up, we started thinking about other type of indirect genetic effect. And one motivation, sorry, I cannot advance the slides. Thank you. One motivation is from this paper, because genetic nurture is actually not the only type of social genetic effect. In this paper by Yasmin Wirtz and colleagues, they convincingly showed that children's genetics can actually affect how the mother treats them. So that they showed that children's polygenetic score can explain the mother's parenting behavior even after you account for mother's genetics. So then we started thinking about, can you sort of generalize this idea to other type of social genetic effect? So more specifically, it's illustrated on the graph on the right-hand side. Basically, if we're talking about a disease in a parent, for example, the Y here can be a disease outcome, then obviously there can be a direct effect, right? For example, the father's genotype can cause a disease risk, or this disease could be affected by children's genotype, could also be affected by spousal genotype. So there are different type of indirect genetic effect. Then turns out it's a very similar problem compared to the one I showed you before. All you need to do, because you don't have that many families in cohorts, all you need to do is to run 3 separate GWAS. One is to run a standard GWAS by regressing everybody's phenotype on their own genotype. The second one is to regress parents' genotype on parents' phenotype on children's genotype. The third one is to regress someone's phenotype on the spousal, on the partner's genotype. If you have that, you can do linear algebra and estimate the red interesting parameters you really want to estimate. Still, then we thought about using Alzheimer as a trait to, next slide, please. To illustrate this idea, we want to talk about Alzheimer's because we, in my group, we actually focus on Alzheimer's quite a bit, and also Alzheimer's is one of the phenotypes where in UK Biobank, people actually report parental history. So you actually know their parents' disease outcome. Right? But then very quickly, we realized that we don't have the third one. You don't have people's spouses' Alzheimer's outcome. So it turns out it's actually a more difficult problem than we initially envisioned. So they had to come up with an approach to solve this problem. The solution we thought of is really that you can actually impute parents' genotypes using 4 sibling pairs in the UK Biobank. If you do that, basically you can run something that's similar to a spousal GWAS that's highlighted by this red circle here. Basically, you have parents' phenotype, because in UK Biogram Bank, people report their parental history of Alzheimer's. And now you also have some average parental genotype. You don't have the spousal genotype, but you have the average of father and mother. But if you run a GWAS there, we call it GWAS-P, you did the algebras, basically you need to change the framework a little bit, but you can still reconstruct the direct and indirect parameters you really want to estimate. So that's the intuition. So we tried it on Alzheimer, but the critical limitation is this really relies on parental genotype imputation, and sample size is a critical issue because you only impute parents when there is a forcibly pair. So sample size here is substantially smaller than other GWAS we use as input. Then next slide, please. No. And so here is what the direct effect estimates look like. It's actually pretty much a standard Alzheimer GWAS. Effective sample size is not very big. Only 9,000, but you can clearly see the ApoE effect. Another GMI significant hit is also from the MS4A locus. It's a well-known GWAS causal locus for Alzheimer's. Perfect QQ plot. You can estimate genetic correlation with everything else. It's underpowered because of limited sample size, but at least it's genetic correlated with Alzheimer's, right? But that's not so interesting. The interesting part about this is the genetic correlation is certainly not 1. Sorry that the scale is actually marked, is blocked, but it's around 75%. So it's not the direct effect estimate for Alzheimer's disease does not have a perfect correlation with Alzheimer's case control GWAS. This is a piece of evidence that says that indirect effect might have a non-trivial contribution. We also did this for spousal indirect effect and also children's indirect effect. Those GWAS are massively underpowered. That's a critical limitation of our current analysis. You can see the effective sample size is only 3,000 and 1,000. Those are tiny sample sizes for GWAS like this. But at least the results are reasonable. You just get no result. But it's not just the power issue, though. If you look at the ApoE effect, for example, in the standard case control GWAS for Alzheimer's, this is the effect size in negative 1.2. You essentially get a 0 p-value because it's such a large effect. Then the direct effect estimate is very close to that. It's negative 1.19. You still get a significant p-value. That's the ApoE peak we saw on the previous slide. And the spousal effect and children's effect are just substantially lower. Not just about the inflated standard error, but also the point estimate are just substantially lower. So then, so basically this is results on Alzheimer's we got so far. But what I want to highlight is basically we have expanded the framework of donuts we introduced before to estimate genetic nurture into estimate other type of indirect social genetic effects, including, for example, how children's genotype affect parents' phenotype. And there are some critical limitations. For example, here, we're not really talking about spouses. We're talking about the biological parent of the children. Depending on what phenotype you want to study, sometimes this actually this actually could matter. And also there could be measurement error in both the imputed parent's genotype and also in reported parental history. This could be inaccurately assessed. But to me, I think so far the biggest limitation, at least in the analysis of Alzheimer's, is because of we really have limited statistical power, especially in the GWAS-P, because it relies on parental genotype imputation. However, the framework doesn't have to be only applied to Alzheimer's. You can use this to other phenotypes, especially when a GWAS spousal effect or GWAS-P is more well-powered, or maybe in some cases you can ignore that kind of effect. You're reasonable to assume the real spousal effect is 0, then you can possibly use this to get, for example, the children indirect effect that you want to estimate. So I'll stop here, and thank you very much.